So we are discussing about biosafety cabinets. A biosafety cabinet has its origins in research in microbiology. So actually they are conventionally found in microbiology labs, and their main intention or main aim of using biosafety cabinet is to protect the person who is working in the cabinet from any contaminating pathogens. So as the name implies, biosafety cabinet, it's basically for safety of the person who is working with any kind of samples, biological samples, from contamination and possible hazard from pathogens. So if you see the equipment, we have an emblem here, yellow color. So this is usually the sign for biohazard. So what this emblem means is that this equipment, it does not mean that this equipment is a biohazard. What it means is that this equipment deals with things, is used to deal with stuff that may potentially be a biohazard. So it is warning people. Suppose a new person enters the lab, he does not know anything, he is just visiting the lab, he simply goes and he is a very inquisitive type, let us say curious person. He goes ahead and touches things, opens things and all. For such a person, this should serve as a warning that okay, please be careful with this equipment and do not open or close it at your whim and fancy. So they should be very careful. So this emblem is for that. Now biosafety cabinet. So on similar lines of biosafety cabinet, you can also talk about another equipment called fume hood. Fume hood, as the name implies, what it does is, suppose we are doing some experiments. So let us say generally if you say chemical biological experiments, while you do the experiment and while the samples in the experiment interact with each other, they will generate some chemicals, mostly in the vapor form. So these chemicals have to be taken out from the immediate surrounding. A fume hood will do only that. A fume hood, what it does is, it will suck out these chemicals and then expose it off. A biosafety cabinet on the other hand does much more than that. As per a US standard, biosafety levels are classified into different types, like class 1, class 2, class 3, etc. and there are set guidelines for each of these biosafety levels. As per each of these biosafety levels, there are also different types of biosafety cabinets also. So this is, so these biosafety level or BSL, biosafety level, by BSL, and the classes are called 1, 2, 3. So it is called BSL 1, BSL 2, BSL 3, like that, BSL 1, BSL 2, BSL 3. So this biosafety cabinet or biosafety hood is a BSL 2 equipment, biosafety level class 2 equipment. What are these classes? I will get into it shortly. Now, one of the main things is to make sure that there are two things. There are two things to make sure. One is a person works on a sample. There are two things. One is the person should not get affected by anything contagious or dangerous in the sample. Another thing is, while the person works on it, the sample which he works on must also not get contaminated or disturbed by the surrounding environment. Depending on these two criterion, also these biosafety class levels are defined. We will get into these shortly. We have understood the basics of biosafety cabinets, what are the different types of containment facilities available. Now let us look at actually what is this biosafety cabinet or biosafety hood all about. So if you can see, this is the control part of this whole biosafety cabinet. We have powered it on. Okay. So now this is, as I have told, this is for a BSL2 environment. So there is enough levels of protection that needs to be taken care of and precautions that need to be done before we even start using it, to be exact. So there are few things that are provided on the biosafety cabinet that we need to do before we start using it. So if you see the knobs, there are 1, 2, 3, 4, 5 knobs provided. Okay. Major ones are, say, there is a UV knob at the middle. That is before we start using it, or actually whenever we do not use it, we need to have the UV light on. So if you see, the UV light has come on, the color inside has changed. What does this UV light do? Whenever we are not using the biosafety cabinet, UV light should be on so that it will sterilize and kill any pathogenic growth or bacterial growth that might happen inside the biosafety cabinet, so that sterility or the sterile environment is maintained sufficiently well throughout the inside the biosafety cabinet at all times whenever we are not using it. But as you all know, UV light, we should not get exposed to UV light. So this biosafety cabinet provides that protection also. Ideally what we should do is that as and when we start using it, we should switch off the UV light. So we should switch off the UV light and use it. But then suppose we forget about it and we start using it. To start using it, we need to lift the lid. This is a glass lid that is provided for our applications. So ideally we should be switching off the UV light like this. So let me switch it off. We will switch it off and then we have to lift this lid, lift the lid and work. You should work like this inside. But then, and you make sure that please never lift the lid fully. This lid is not meant for that. Why this lid is provided has a reason. Everything has a reason. So we should keep only at our working height and then work. Why this is so? This is because suppose something happens there, some chemicals get released from there, they should not reach our eyes. So that protection is provided by this glass. That protection is provided by this glass, and you can safely work with only your hands inside, and you can reach, and the cabinet size is such that you can reach throughout the region of the cabinet. Hope this is clear. Now let's see. Let's get back to our discussion. Suppose now our UV light is on. Let me switch on the UV light again. So our UV light is on again, but we forgot to switch it off before we started. Then we are starting to use. Okay, please look here. We are starting to use, and I am lifting the lid. Immediately the UV light goes off. So this is a protection provided because you should not get exposed to UV light. It can cause diseases like cancer. So you have to be very careful that you are not exposed to UV light. So the cabinet gives that protection also. Any biosafety cabinet that works in BSL2 facility will have these features installed. Let us see it again. So let's say I try to switch on the UV light while the lid is up. That also that equipment will not allow you to switch it on. See, the lid is up right now. This gap is there. I am trying to switch on. It is not switching on. Now I am closing it. Now it's closed. If I switch on, the UV light will come. Let's say we try to open it. The moment it crosses a very small area itself, the light will switch off. So this is a safety precaution that is provided in case we forget to switch off the UV light. Now we understood about the UV light, right? There is another. The second option that is provided is for normal visible light. So we can see it now here. The second option is for normal visible light. We will shortly see the normal visible light, and then the first knob is for circulation, fan and exhaust. So before we start using the biosafety cabinet, the protocol is that we should always have the UV light on. The UV light is on. This fan should be running. So when you switch on the fan, the alarm will ring here. The alarm is ringing because it needs some time to reach that circulation speed. So it is telling that I have not reached my set speed, so please wait. So I can actually, because I know that this is the issue, we can actually mute the alarm. So the alarm will go. So if any other unexpected incident happens also, this alarm will ring. So there is a circulation happening inside. So this circulation will constantly flush in good quality air through the hood, and also take out any chemicals that get secreted. Now so while you work on the hood, it is advised that always you have the white light on. So let us switch on the white light. So I have switched off the fan because now it has been properly cleansed. So you can work without the fan running. So what we can do is, once your work is over, you can put on the UV light, put on the fan and leave it. So that will sterilize the environment inside. Now we have switched on the working light. This is the working light. Now with the working light on, we can lift the lid, keep our hands inside and work. This way we can work. This is how we should work in the biosafety cabinet. Now I think it is very much visible now inside. These knobs, see these two inlets are for water, distilled water and any other chemical or circulation that you need. Water you can bring in, even air at high pressure air, if you want to clean some wafers with nitrogen gas, that also you can bring in. That is your own wish. You can always connect things in the other side and bring them here. Now here you have a pressure indicator showing what is the pressure inside the hood right now. So right now it is showing the default atmospheric pressure only. Now let me open it fully. The cabinet also provides 2 plug points of maximum output of 220 volt 3 amps in case. So what happens is, while we work we will have to use other equipments also, other equipments like centrifuge, cytocentrifuge and let us say hot plate. So these hot plates, cytocentrifuges etc. have to be kept inside, right, but we have to power them also. So for powering those equipments, these plug points are also provided. Now the tube here which you see here is a UV source. So this will generate the UV light source. Our white light source is kept inside here. It is there in these areas and the light comes or falls on it. The light is not kept there because you will not get the glare from that light. So light comes from here, it floods the biosafety cabinet nicely and properly. So let us close this again. So especially when you are working with, so these are all equipment level protections that are provided, right. These are anyway man made. So you cannot always be very sure that these equipments may not malfunction. So they may malfunction at different points. So always wear any UV protection glasses with you, like I am wearing now, so that in case this protection mechanism doesn't work, so that UV light is on, you can see the UV light there. So but by default this protection is there. But in case the protection stops working because of some control circuit failure inside the equipment, you should still be protected. So that is why you should wear UV protection glasses. Again, if you see the glass bottom, they are also providing proper cautions. See here. So this is at the bottom of this glass. So caution, danger, protect eyes while UV light is in use. So even through this glass, they are advising us that we should use UV light to protect us, because anyway even though we are not exposed to it, we are actually seeing the UV light, right. So they are asking us to protect us from the UV light. And for this table, this is a SUS table. So for this SUS table also they are giving caution, like the SUS table must be cleaned up while it contacts with caustic medium or the work is finished. So every time you finish the work, you should clean up this table with nice distilled water or even acetone or IPA. You can wipe the surface, clean it either when work is finished or while you are using any caustic medium. And this is the safety limit that is provided. So these are the major features that are available inside a biosafety cabinet. Shortly I will show you, after keeping some basic biology equipment inside, how we have to actually work with things inside. So one more thing, if you come up, there is one more knob provided here, which is the fourth knob, that is power socket knob. So what this does is that, so we have seen that there are 2 power sockets right inside. We may not need that power on that sockets all the time. So we can actually switch this on to make sure that power comes there, otherwise power need not come there. That is the whole idea. So that power is separately provided, that we need not separately power those sockets. From the power line that powers this cabinet, those sockets will be powered, and you can actually switch off power output from those sockets through this option. And now you see that this is the working area, right. This much is only the working area. Then what is this much area? So this is the fume hood that is provided, that processes whatever fumes are coming from the sample. It will clean that fumes, remove all the contaminating equipment, and then exhaust it out to the environment. So the air that comes out of the environment will be free of any contaminant or pathogenic material. That is what is there in this much area above the biosafety cabinet. Now let us look at the manufacturers themselves. As per their guidelines of manufacturing, they have provided few operation guides. We will just quickly go through what are the operation guides that they have provided. It will give us a better idea about how to use the biosafety cabinet over and above what I have just described now. So what they are telling, we will quickly go over them. First one, put the sliding sash on height of 200 mm before starting the fan, and keep fan running for 15 minutes before using the cabinet. This is what I had told. So before we start using the cabinet, you should switch on the fan, keep it running for some, you should switch on the fan, keep it running for some time, so that the air within the cabinet is purified and the ultraviolet light also cleanses the environment. Say like this. So I have switched on the fan, it will cleanse the environment. So if you keep your hand inside, I have switched on the fan, so these vents are there here, from there air is coming. So if you look at this paper, you will feel that the air is coming, see. Actually it is that whatever is air is inside, right, it is being sucked in at this region and it is recirculated from another portion. From here it is getting sucked and it gets recirculated from top. So this circulation is maintained here. So why? Because it is sucking the air from here, we need to close it so that properly, so that it can cleanse the inside properly. Then next one, please be gentle when sliding the sash. So what is sash? Sash is this glass window that we know, that is called the sash. So it is we have to handle it gently, because once or two times when I was pushing it up, it is going away. So it will go and hit in a slightly fast way. So you should make sure that you are sliding it up very slowly. Keep your hands on the slide, don't leave it like push it and leave it. Keep it in your hand, slide it till the top. While you are using it, you will not have to slide it till the top, mind it. This is only when you want to clean the area. Otherwise, you will use it only with your working distance, working height. Keep it like this. Sit on your chair in a comfortable position. Keep your feet nicely inside. Put only your hands inside and then work. Once your work is finished, slide the door closed, switch on the UV light and leave. Before using, switch on the circulation, clean the air inside, switch on the UV light, sterilize, then use. This is the protocol. Now next one, try to operate or put articles in the central of the working area and do not block the return air grills. We had seen the air grills, so we should not put anything on top of those grills. We saw the grills where air is being sucked, right? Don't put any equipment on top of it. Once you start using it, this table will get consumed by lot of equipment and samples. While you do that also, make sure that you don't clog the entry points or exit points of the air. Next, minimize articles or instruments putting into the cabinet and do not disturb the air circulation. This is again a repetition of the point where we told, even though we might have to use some equipment inside, try to keep it minimum, and the things that you can do outside the biosafety cabinet, the less pathogenic or non-pathogenic work, you can do outside the biosafety cabinet. Next, articles should be decontaminated before putting into the working chamber. This is a very important fact, because so suppose we will be using different types of samples, equipment, different devices, etc. inside the biosafety cabinet. If we don't sterilize them properly, whatever protection we are providing through this very costly equipment will come to naught or zero if our equipment or the tools that we are using inside the cabinet are not sterile. So we have to as much as possible the items that can be sterilized through equipment called autoclave, which we will see shortly, should be autoclaved. Other big equipments should be properly sterilized and cleaned before they are used inside the hood. That is what they are telling. Next, minimize the activity around the cabinet, like arm in out the cabinet. The arm movement should be slowly to avoid the disrupting the airflow. This is self-explanatory. Next, do not stop the fan during normal operation. So the fan should always be running. Don't abruptly stop the fan. Then, to ensure the safety of your articles, move them out of the cabinet in time after operation is finished. Keep the cabinet running for 15 more minutes after operation finishes for full self purification purpose, then turn off the fan. So the fan should be running for at least 15 minutes. So let this siren go off. Let's see if the siren stops after some time. Now I have muted it, because for 15 minutes the siren will run, because it is telling us that it is purifying. Then UV light can only be on when fan is stopped. We have seen it. If I try to switch on the UV light when the fan is on, it will not switch on. See, I am trying to switch on, it will not switch on. UV light can only be on when fan is stopped, sliding sash is closed and the light is off. When all these situations are satisfied, only then can the UV light be switched on. When all the sash or the sliding sash should be closed, the fan should be off, then only the UV light can be switched on. Keep away from the UV light when it is on, so as not to cause injury of skin and eyes. Clean the drain, spillage, trough and work table frequently. Please, we have to keep this area clean. Please choose neutral detergent and avoid chlorined cleaners, because they can react with the surfaces that are there inside the biosafety cabinet and cause corrosion. Choose proper disinfectant according to the articles operated in the cabinet and clean up the residuals of the disinfectant with clean water in time. So it is very important that we use very clean water, preferably distilled water or DI water, which is even better, which is called deionized water. Such a water will have no ionic content in it, so it will not be reactive even to a very small extent. By default also water is not that reactive, but it will still have lot of dissolved salts and ions in it. So we have to use deionized or distilled water, so that any unknown ions dissolved in the water don't react with your sample or your cabinet. If pipeline system needed, please connect air supply and exhaust pipe correctly and ensure the enough compensatory fresh air into the room. For that, we make sure that we have our HEPA filter arrangements, which will constantly push in very clean air into the room. Please refer to the user manual for details of fumigation and sterilization. Sterilization we have told you about. So fumigation will be carried out by this top part. What is fumigation? Fuming. So whatever exhaust chemicals that come out of the sample, they will be burnt. That gases will be burnt again, so that they are sterilized. So this is sterilization through burning. Fumigation is that. Let's say you have cut down some trees and they are actually going bad. You actually burn them, right? So that is fumigation. So fumigation is sterilization through burning, fumigation and sterilization. Then strictly forbidden to demount or refit the electrical system without the manufacturer's permissions. Okay. These are the major precautionary measures that you should take care. Shortly, we will show you how to use a few equipments inside the biosafety cabinet, so you get a hang of it. These biosafety levels we will get into shortly. Before that, I have mentioned that biosafety cabinet is used mainly for protection of the worker or the researcher from pathogens. So pathogen is a very generic word. What are pathogens? Are any foreign body that tries to get into your body, that is a pathogen. That could be a harmful bacteria, that could be a virus, that could be a protozoa. There are several protozoic diseases also. So we need to protect ourselves while we are working from these harmful foreign bodies. So biosafety cabinet is mainly used for that. Now, a class 1 biosafety cabinet will provide protection. So there are three different types of protection. Personal protection. Personal is us, the person who is working on the cabinet. That is personal protection. 2 is environmental protection. That is protection from harmful agents of the environment that I am or anybody is working on. That is this area and this lab in general, and also to the building or surrounding places. And the third protection is a product protection. Product protection is a protection of the sample which we are working on from being contaminated or changed by factors not under our control. So, we should not, if it is something that we have introduced and we want that sample to change, that is different. But suppose the environment itself is making the sample to change in a way that we do not desire it, then that product is not protected, but we need to protect the product from it. So, that is called product protection. So, 3 main protections are there: personal protection, environmental protection and product protection. Okay. So, a class 1 biosafety cabinet provides basically personal protection and environmental protection. Product protection is not provided. What it means is that the air that is used, so in this cabinet you can anyway see, so there is a enclosed area inside, right. So, you can actually if you see inside, there are 2 nozzles. So, I will just lift it now. So, there are 2 nozzles. So, these nozzles can be used to supply deionized water or distilled water, very clear water, and also to circulate air inside the biosafety cabinet. Now, in a class 1 biosafety cabinet, as I told, there is no product protection provided. So, what it means is that the air that comes in contact with the sample might not necessarily be free of contaminants. That is what it means. What it also means is that you will just be circulating a normal air within your cabinet. So, that is class 1. A class 2 biosafety cabinet. Class 2 biosafety cabinet would include BSL 2 and BSL 3, biosafety level 2 and 3. A class 2 biosafety cabinet would provide all 3 types of protection, that is personal protection, environmental protection and product protection. How it does this? It does this through HEPA filters. We have already covered what HEPA filters are, what are tower modules. We have tower modules in this lab, we have seen it. So, what HEPA filters do is, first of all, the air that gets circulated within the biosafety cabinet would themselves be HEPA filtered. That means HEPA is what? High efficiency particulate air. The number of particulate matter greater than 5 microns is very much controlled in a HEPA filtered environment. So, if it is a class 1000 clean room, that means there will be only 1000 number of particles greater than 5 micron size within your enclosed space. Class 10 clean room would mean there will be only 10 particles. So, you have huge amount of control in the quality of air that you would be circulating in your biosafety cabinet. So, when your biosafety cabinet has provision to circulate HEPA filtered air and also the chemicals that come out while your sample interacts with the chemicals that you add, the vapors, these vapors will also be HEPA filtered before it enters the environment. This way it provides environmental protection and personal protection, and because we are using HEPA filtered air, very clean air only to circulate around the sample is also protected from unwanted changes. Thereby we get, yes, product protection. So, product protection, personal protection and environmental protection. So, this is class 2. So, class 3 is a very special type of biosafety cabinet. This is not that, they are mainly used for BSL 4 containment. So, see, if you see, there is this class 1, class 2, class 3 is a separate US government or a US standard based classification. Parallelly, we have the biosafety level classifications also. So, biosafety level 1, 2, 3, 4 up to 4 are there. What is biosafety level 4? Biosafety level 4 are applications where we have to use highly guarded containment facilities for very, very dangerous viruses and contaminants like HIV virus, Ebola virus. So, such very, very, very dangerous viruses, if you want to handle, you need to use a class 3 or a BSL 4 environment. There the protections provided are much more in detail. We can go to the details of it at a later time. So, you just understand that there is class 1, class 2, different types of classes of biosafety cabinets, and their specific applications are for different purposes. And how they are differing is through the arrangement of the air and the extraction of fumes that come out, how they are processed before they are put back into the environment. So, BSL 4 is the highest level of protection, because once you have to be very careful while handling such infectious agents. Now, we will look at some of the knobs that are present in the equipment. So 100 microliter it has been set. Now let's see how this operates actually. So there are basically two knobs in this. This two violet you have to see. So this is where you take out the sample. So here, if you closely observe my hand, I press it. This will create vacuum here. Then when I take it back, it will take exactly whatever volume we have shown right here. In that much volume it will take in the pipette tip. It's called tip. I will show the pipette tip shortly. So like this. Then after this you can actually, if you are transferring, so let's say we take it and we are transferring it somewhere else. If that is the case, if you want to finally transfer all the droplets, you have to push it further like this. There is an extra push that can be provided. That is only for transferring whatever is there already in the tip back to the second container. But the volume measurement will be through this only. Don't push it further like this. What is this used for? This is used for finally discarding the tip like this. So let me show you how a pipette tip looks like. So this is a micro pipette tip box. So tips can be arranged in this box. So tips. So this is how a micro pipette tip looks like. So this is the 200 microliter. So it can hold maximum 200 microliter of sample volume. So now how to take this tip. So you take your micro pipette, put it in the hand you are comfortable with, put it like this, give few taps, and your tip is already engaged into your micro pipette. Now if you have to take sample volumes, you have to press it like this, and then it will take the sample volume, and it will then you can go and release it somewhere else. Now let us say we have to remove it. So ideally you will be handling cell culture suspensions, right, that you might have to transfer from one dish to another. Let us say at that point you do not want to actually touch this thing, the tip. So what you do is you will do everything like this. You will transfer it from there and then transfer it back, and then there will be a waste box kept there or below, just below your here or just below your biosafety cabinet. And then what we will do, just press this and the tip will fall into the waste box. I will just show it again. I put it in the tip box. So I am taking a tip. So tip is tightly put. We will take samples, we will transfer it, and then we will discard it like this. Okay. Now let us see how actually sample volumes change. Now let us see how we are actually transferring sample volumes. See, I have my pipette tip. So what I have here is a culture of cells in this dish, which is purple in color. The cells are in suspension, that is they are suspended in a medium which is DMEM, which is Dubois minimum essential medium. This medium gives the necessary nutrients required for the cells to continuously grow. How the cells are grown using a CO2 incubator, we will introduce to you soon. So this is a dish containing cultured cells. Okay. So now I want to transfer 100 microliter of these cells to this dish. How do I do that? So I will first show you how much the sample volume differs when we take 200 microliter and 100 microliter. Let me just adjust it back to 200 microliter, which is the highest volume. So as you can see, it is 200 microliter now. So I will now take, so you see how I am using, only my hands are inside and the hood is almost closed, so that my face is protected by this glass shield. Now I take a tip, I open the lid of the dish. Now, so I press it. So when I press and then I introduce and release, it will take in the sample volumes. So I have kept the other dish ready. So if I release it, you can see that 200 microliter is taken. You can see it in the tip, but I don't want 200 microliter. So let me put it back like by pushing it like this. Okay. Now see, one drop is hanging. Now to drop that drop only, we have to press it further. So that drop will go. Now I don't want 200 microliter. You guys saw that the volume came till here. But I want to transfer only 100 microliter. So let us reduce it. So now I have set it to 100 microliter. Now let me see how much volume comes in the tip. See, earlier it had come till here. Now it has come only till here. That means only 100 microliter is transferred here. Let me show it to you again. Let me show it to you with very less volume. Let me show it to you with 20 microliter, let's say. So I am reducing it further. So let's not go to 20, let's go to 50 microliter so that it is clearly visible to you. Now I have kept it to 50 microliter. Now I am taking 50 microliter only. So we have seen how to take 100 microliter, 200 microliter, how it came in the pipette tip. Now as I had shown, we have set it to 50 microliter. Now let's see how much volume comes when I set it to 50 microliter. See, as you can see, 100 microliter had come till here. This ring is there, no? Till that ring it had come. Now 200 microliter had come till here. Now 50 microliter has come only till here. So that we do not worry about how much volume we have to take. We just need to set it here. Now the liquid also will not flow out. So it is already there. Unless we press it and drop it, it will not go away. It will be inside the tip only. Now let me take it back to 100 microliter. So this is a suspension of cells in TMEM, as I had told before. Now I have set it to 100 microliter here, 100. So we are taking it, 100 microliter is taken. This is the next dish to which we have to transfer it. So I am transferring it. See, make sure that all drops within the tip are dropped. If they are not dropped, please make sure that it is dropped by pressing it further. So 100 microliter measured volume is now transferred to this dish. Hundred microliter is now transferred. See, it is 100 here. We have transferred from here to here 100 microliter. Let's say if we want to transfer another 100 microliter, I will just press here, take it, 100 microliter is taken, just come to this dish, press it, 100 microliter is transferred. So two times we have transferred 100 microliter to this dish. So we have 200 microliter in this dish, 200 microliter or 0.2 ml. Now the use of this tip is over. For each and every time you have to use a fresh tip, because as you have seen, it is very easy for this tip to get contaminated. These tips, micropipette tips, are not meant for reuse. As soon as you use it, you have to throw it away. So we have to discard it later. So like that we should keep it aside or discard it in a discard box next to our place, as far away from our experiment as possible. Finally, you have to take it out and clean the place with tissue paper and maybe acetone or IPA or ethanol, some alcoholic solution. This is how pipetting is done in tissue culture dishes. These are, as I had shown before, these are the same 35 mm dishes we had seen when we were studying the inverted microscope. This is the tip box where you can arrange tips so that you can easily take them out, so that your use of your hands should be avoided. This what I am doing right now, this should never be done. This is banned inside a biological lab. Should never do this. You should do like this. Best practice. I am showing it to show you that you should not do it. Best lab practices, you will have a tip box. Your tips will be there. You take your pipette, take it, do your experiments. Minimum involvement of your hand. No contact with the tip, because tip carries the samples. No, neither the sample should get contaminated, nor should you get any kind of pathogenic infection from the sample, because you don't know, the sample might have viruses, HIV virus, Ebola virus, anything could be there. So this is how you should handle tips. Next, I will just show you few other equipment that are used. This is just like we saw micro pipette tip, which is this one. This is a 2 ml bigger pipette tip. This is not a micro pipette, this is just normal pipette tip, 2 ml micro pipette tip. So, in case you want to transfer huge volumes, you can use this with another equipment called a Eppendorf pipette. That is a different pipette, which we will show to you later. So, this is another 2 ml tip. You can see that volumes are marked here nicely. This is Borosil, made up of Borosil glass. Now, to transfer, so if you want to centrifuge your cells, like centrifuging is making your cells come down and supernatant to be separated, just so that you can separate only the cells out. You can use tubes like this. So, this is 15 ml tube. This is 50 ml tube, as you can see. So, 15 ml tube, 50 ml tube. So, this can be used for varied purposes, and for this conical part also there are what you call sub calibrations, so that you know what you are taking. Here also for the conical part you have sub calibrations. So, this is starts at 2 ml here, this will be 1.5 ml, and within 1.5 ml also you have sub calibrations here. See 1.5 up to 0.1 you can actually measure out in this tube. So, students, I think now it is clear to you how you should use the biosafety cabinet, how you should keep minimum parts of your body inside, and how to use this. This is the crux of how biosafety cabinet is. At later point in the course, in case we are doing any other further experiments, those experiments also we will show in the biosafety cabinet.